Okay, our next speaker is Dr. Hatem Sabaawy. Hatem was recruited to the Canton Institute in New Jersey last year when we needed a sushi chef. So he received his training, medical and graduate training at the Cairo University Hospitals in Cairo and the New York Medical College. And he's a former fellow of NIH NCI Transplantation Immunology branch of the Center for Cancer Research. And he's going to talk to us today about stem cells or sushi, zebrafish as a leukemia and stem cell transplant model. Hatem?
Thank you, Dr. Bertino, for the kind introduction, and I'm deeply thankful to the organizers for the invitation. It's actually a great honor to be talking to you today, especially after such a distinguished panel. What I'll try to do is to hopefully convince you that zebrafish is not just a great development model, but actually an evolving cancer and stem cell transplant model. So you might heard about zebrafish in the news when 2 years ago scientists from Penn found that or used zebrafish to unlock the mystery of our skin color when they identified an ion channel that's responsible for the golden color of fish is also responsible for the fair skin color in humans. And this was dated back to migration of humans from Africa. You might have seen zebrafish embryos on the cover of Science or other magazines, but what is definite is that zebrafish will impact cancer research. If you look for zebrafish as a hematopoiesis model, if you actually do a blood smear from fish and you look at the phenotype of the cells displayed here compared to human cells, you see the highly conserved morphology of these zebrafish cells. With the exception of nucleated red blood cells and nucleated platelets, the morphology is about the same. Their immune system is also conserved. They have conserved cytokines and complement cytotoxic effectors. They have a RAG-dependent immune system with all the major components of B, T, and K cells, enzymes as TDT. They have a high diversity of rearranged T cell receptors and all the TGF-beta components. Yeah, I'm obviously not handling this well. So zebrafish will have a great impact on cancer biology and stem cell biology, and this slide summarizes that. Why do we like to use zebrafish? You can house large number of fish in a small space. Mating of a pair of fish will produce 200 embryos a day, and you can repeat this weekly or bi-weekly. The cost of maintenance is relatively low compared to mouse maintenance. You can do genetics and genomics in fish much easier than in mouse. That includes transgenesis lineage tracing. We have a resource of more than 3,000 available mutants from University of Oregon, and you can acquire these and do your assays on them. There is also an insertion and mutagenesis database based on retroviral insertion, and hopefully in 2 years, all the zebrafish genes will be saturated, and there will be available insertional mutants or what you would call knockout fish for almost every zebrafish gene. You can do carcinogenesis easily in fish by aqueous exposure. Simply you put in the carcinogen into the plate, it diffuses through the water and goes into the fish, and you can see the effect. When they do develop tumors, these tumors are very much similar in histology to mammalian tumors. They have the same conserved cancer gene pathways, angiogenesis, and several cancer-prone lines have been developed that will be subjected to modifier screens. Earlier this year, the lab of Lin Zhang in Boston generated a transparent zebrafish displayed here on the top. And this will be a great resource for using it for studying stem cell development. Yeah, if I can get this to move. Okay, so the first part of the talk, I'll describe to you our leukemia model. It's based on a fusion protein found in about 25 to 30% of ALL patients. And I will briefly discuss with you our preliminary data on using fish as a stem cell transplant model. TEL-AML1 is a product of the T1221 chromosomal translocation. It's found in about 30% of these children with ALL. These children will develop leukemia at a mean age incidence of about 2 to 3 years. A lot of work has done on leukemia in twins, pioneered by Mel Greaves' group in the UK. And they show that these twins actually will exchange the fusion in utero. And one of the twins will have leukemia and the other will not. So the presence of TEL-AML1 is not sufficient to cause leukemia in these kids. If you do a bone marrow aspirate, you will see that their bone marrow, the normal heterogeneous bone marrow, is replaced by those lymphoblasts. So based on the data in the literature, the twin data first from Mel Greaves and others show that this fusion protein develops in utero. It's also found in about 1% of normal cord blood samples without having the direct effect causing leukemia. It occurs before the IGH rearrangement, and long latency is required for leukemia development. What's particularly specific about this fusion protein is that it's only found in pre-BALL. So there is something within the sequence of these two transcription factors or the fusion itself that regulates B-cell lymphopoiesis. The sole transgenic mice that was published from Gilliland's lab, when they use an IGH enhancer promoter to drive TEL-AML1, none of these mice developed leukemias, and they were not able to show any growth factor independence in cell lines. Other mouse transplants models also were published where retroviral LTRs were used to express TEL-AML1 in bone marrow cells. The marrow cells will be transplanted into radiated syngeneic mice. All of these mice develop pre-leukemic condition, and only in one case, actually, from Hopkins, I believe Dr. Seven was involved in the work, this leukemia incidence increased in the presence of additional mutations. So when we decided to develop transgenic zebrafish, we looked at B-cell development, and we tried to target this fusion to the potential leukemic stem cell. If you look at this displays actually the early development of B cells from a stem or a progenitor cell to pre-B cells to immature B and mature B cells. So the genetic hit may occur at the stem cell level, and then cells will arrest at the pre-B cell stage. The alternative theory is that all the genetic hits will occur at the pre-B cell stage, and this will lead to accumulation of cells arrested at the pre-B cell stage. So the potential targets for stem cells are within these stages of B-cell differentiation. We decided to target these two stages. First, target the early cells, the stem cells, using ubiquitous promoters that essentially drive in the stem cells as well as other cells, or target the pre-B cell stage with the RAG2 promoter. We developed several transgenic lines based on expression from zebrafish-specific promoters. Displayed here are our constructs that were used: the actin promoter alone or fused to GFP, the Xenopus elongation factor promoter, and we identified about 500 expressing transgenic fish compared to a lymphoid-specific expression from the RAG2 promoter with a larger number of fish. We screen these fish by doing blood smears to detect early leukemias. When there are signs of leukemias, they are sectioned and analyzed. If you develop a transgenic fish with a fusion to GFP or DS-RAD, you can actually detect it the next day. Simply you inject the fish. The next day you put them on the fluorescent scope and you pick up the green fish. This is your transgenic fish. The not green fish are the wild-type sibling. So it's a great model for doing transgenesis, and the expression of TEL-AML1 in these fish led to phenotypic changes. This is the marrow of the fish. Their marrow is actually in the kidney. You would see a section where there is a cross-section in a tubule. The intertubular tissue is the marrow, where the hematopoietic cells are. And this smear shows marked enlargement in the stem cell population of the TEL-AML1 fish in panel A compared to wild-type fish. We separated these cells and subjected them in panel B to a mitogen-induced B-cell clonogenic assay to see if these cells would actually be able to mature and differentiate into B cells, as I demonstrated in the B-cell differentiation scheme. And indeed, only the EGFP control cells will develop B-cell colonies, but those expressing TEL-AML1 stayed at the single cell level and failed to induce B-cell differentiation, indicating that this fusion causes a differentiation arrest at the pre-B cell stage. In about 5%, 3 to 5% of these transgenic fish, they develop acute lymphoblastic leukemia that's widespread. The average latency is about 8 months, and these leukemia cells, if you look at panel A, the arrows indicate the normal kidney marrow size, and the arrows here indicate the leukemic fish with marked expansion in their kidney marrow that's all infiltrated by acute lymphoblastic leukemia cells. These cells spread to all the organs, and they stained by immune histochemistry positive for GFP that's fused to TEL-AML1. When you separate these cells and subject them to a dual myeloperoxidase periodic acid shift staining, they stain negative for this compared to the 1 to 2% normal cells that were positive on the same smear. These leukemias are 98 to 99% blastic cells in the peripheral blood. They were all negative for the stain, indicating that they are lymphoid in nature and they are neither myeloid nor erythroleukemic in nature. As I mentioned earlier, only when you use ubiquitous promoters, there is 3 to 5% incidence of leukemia or lymphoid hyperplasia, but not when you direct the leukemic transformation to the RAG2 expressing cells. This leukemia was transplantable to wild-type recipients. So when we condition recipients with sublethal irradiation of 25 grays, we can transplant these cells, and all the recipients will develop ALL in 5 to 8 weeks in a phenotype very much similar to the donor leukemias. To look more into the origin of these leukemic cells and how leukemia developed in these fish, this slide displays the normal definitive hematopoiesis with the transcription factors involved in it. And since TEL-AML1 is involved with pre-B-ALL, we thought that by studying the expression of these markers, SCL as a marker for the hematopoietic stem cells, Ikaros as a marker for a lymphoid progenitor, and the differentiation markers IgM and TCR, this will give us an idea on where did this leukemia started. Indeed, we did an RT-PCR on 10 leukemic cells followed by southern blotting compared to normal controls, and displayed here are 5 leukemic fish, samples from 5 leukemic fish, showing that all these leukemias are negative for IgM, negative for T-cell receptor. They all express the hallmark of pre-B-ALL, the neutral endopeptidase zebrafish homologue of CD10. 3 of the 5 express RAG2, despite the fact that when we use the RAG2 promoter, it never induced the leukemia. they all expressed the common lymphoid marker ECROS and the stem cell marker SCL. This indicates that this leukemia mimics the phenotype of pre-B ALL in humans. We further analyzed the association of different genetic hits that occur in these transgenic fish. And this was done by developing a consensus based on the microarray data coming from St. Jude and European oncology groups. And we combined these data into a panel of pathway-related genetic changes. We analyzed these changes in the leukemic samples. What's displayed here are those that were significant or display tendency to significance. First, we developed a one-step real-time RT-PCR. This is based on taking 10 leukemic cells from each leukemia fish and comparing them to a range of expression of transcripts for all these genes. So the range is displayed on the left side, and the range of expression is based on expression in wild-type fish, in transgenic fish that did not develop leukemia. So any changes in the transcript level directly correlates with transformation. And what we found significant is that there is a reversal of BCL2 Bex ratio. There is marked drop of the endogenous zebrafish tail, the partner in the fusion, and there is a tendency for loss of INC4, though it's not significant because of the small sample size. Based on these data, we proposed this model that tele-AML1 leukemia actually starts at the hematopoietic stem cell, and it causes differentiation arrest, leading to a pre-leukemic clonality. These cells will stay for a long time. We know that twins are born with these leukemia, or these cells expressing these fusions, though they don't develop leukemia for 5 and 10 years, until they acquire these secondary genetic hits that are affecting cell cycle and apoptosis that they can develop OCAD. We also developed new transgenic lines to confirm this by expressing tele-AML1 with the SCL promoter that drives expression at the stem cell level, and those develop a low incidence of leukemia. Using the zebrafish IGH promoter, none of these fish will develop leukemia. So this actually confirms the leukemic stem cell origin at the hematopoietic stem cell. Based on these data, I would conclude that we established transgenic fish expressing tele-AML1, that leukemia development requires the expression of this fusion in the hematopoietic stem cells, and that tele-AML1 causes B-cell differentiation arrest and requires secondary genetic events within the cell cycle and apoptosis pathways for leukemia development. In the this is actually displaying what we will be doing. It's more like future direction, what we are going to use these transgenic fish for. So now we have a leukemia-prone line that develops or have a low incidence of leukemia, about 3-5%, with a long latency. This is a great tool because you can study the genetic events that happen with an initiating event of tele-AML1. What we're doing now is in collaboration with Paul Lu at NIH, who has a RUNX1 mutant fish. We're crossing these fish, and in collaboration with Lenzon in Boston, we're crossing them with a RAS overexpression fish that develop MDS. We also have identified mutants for RB, INC4, and other genetic markers that are useful for crossing them. We developed transgenics for BCL2, some microRNAs, as Dr. Seven mentioned earlier, that regulate hematopoiesis, and we thought that they might be very well involved with an ALL development since they were also found in B-cell lymphomas. And finally, we're developing a mutagenesis screen based on retrovirus and transposon systems to identify those second hits and the genetic pathways involved in tele-AML1 leukemia. I'm going to shift gears a little bit here and talk about some of our preliminary data on actually using fish as a transplant model for lineage tracing and trying to identify the clonogenic potential of stem cells. So the traditional view for stem cell development is that there is an irreversible switch that cells, the early stem cells, when they become progenitors, they undergo a irreversible change to eliminate their propensity. The graded propensity now is a different concept after several groups, starting from the Kyoto group in Japan, were being able to induce adult cells to become pluripotent cells. So the evolving view is that there are several progenitors in between the activated stem cells and the terminally differentiated cells that might respond to the microenvironment and change phenotype. We're trying to test this theory in zebrafish. So over the course of the last 2 years or so, we developed several tissue-specific, several transgenic zebrafish expressing GFP or DS-RED in a tissue-specific manner. They're listed here. What I am going to show you is briefly some work on the hematopoietic specific expression in these lines in transplant studies. So normally the induction of the hemogenic endothelium by certain factors will lead to the development of angioblasts or hematopoietic stem cells that develop into mature blood cells. This also is controlled by a long list of transcription factors that are hematopoietic specific, and that might also drive the differentiation of B lymphocytes. The concept is if we can genetically label stem cells using specific promoters and then transplant these genetically labeled adult cells in recipients, monitor them live in vivo on a fluorescence scope, we can actually detect the tissue contribution of these stem cells and if they are indeed being able to be pluripotent or change phenotype. If I would start the movie on the left-hand side, this is if you label blood cells in zebrafish, since they are transparent, you can actually monitor it in vivo and see where all these cells are going and what is their contribution. Now, if you take this fish on the left side as a recipient fish and then transplant cells from DS-RED labeled cells, now focus on the area highlighted in red here. If we would play this movie, we would see that there are some DS-RED labeled cells within this recipient. This model will actually allow us to directly and instantly tell the percentage of chimerism between donors and recipients and distinguish donor-derived cells from recipient cells. So we use the promoters of these transgenic fish to drive tissue-specific expression, shown here in these embryos. So the actin is a ubiquitous expression. The blood-specific expression was achieved by SCL, which expresses at the hemangioblast level. The lymphoid-specific expression was done by the RAG2, and the fly is an endothelial marker, so it expresses in blood vessels. These promoter fragments are sufficient to drive expression. This is actually a whole mount in situ hybridization of the wild-type expression of these genes. And what this indicates is that these promoter fragments reflect the GFP expression normally found in these fish. So we grow these fish until they are adults, and then we separated their bone marrow by flow cytometry and transplant them into adult recipients. To remind you again, all the transplants so far are between adult fish and adult recipients. If you put a fish bone marrow sample on a flow cytometry, they separate into 5 different gates. So we sorted out every gate of these and cytospin them on a slide. And what you would see is about 50% of the fish marrow would be erythrocytes, 20% will be the dark green gate with myelomonocytes, 15% are the light green gate with lymphocytes, and about 7% will be in the dark blue gate, the navy blue, with progenitors. So every sorted fraction of these were tested if they would have the stem cell potential. And the way this was done, if you transplant every fraction of these and they would give radioprotection, they would allow the recipient fish to survive radiation, that simply indicates that these cells contain the progenitor fraction or the hematopoietic stem cells. So we transplanted every fraction of these, and only the progenitor fraction provided the expected radioprotection effect. So we took those GFP positive progenitor fraction and we designed 3 experiments. The first one, we want to know what is the number of GFP positive cells in this progenitor fraction. And what would this tell us is each transcription factor promoter, how much does it contribute to the progenitor fraction? In other words, within the stem cell fraction, how many of these 7% are committed to being a lymphoid progenitor, how many are committed to being an endothelial progenitor, and how many are committed to being a stem cell progenitor. This is the assay. If you would focus on the second panel, an overlay of forward and side scatter with a GFP expression. As you would expect, within the actin GFP line, all the cells are green. With the tissue-specific expression in lymphoid progenitors with the RAG2, only the green and the blue gates are GFP positive. To remind you, this is how the separation is on our facts. So with the RAG2 line, you see GFP within the progenitors and the lymphocytes. With the fly line, you see it within the progenitor, and with the SCL within the progenitor, with some differentiating cells. So we sorted out the blue gate, the progenitor fraction, and we looked at the percentage of GFP positive within this fraction. With the actin line, almost all the cells were GFP positive. Within the RAG2 line, 60% of the cells have a lymphoid commitment based on the RAG2 expression. 1/3 were endothelial progenitors, and 1/4 were hematopoietic stem cells. These fractions were sorted out and transplanted into either adult fish or embryos at the blastula stage. So first, the adult cells. If you take GFP positive adult marrow cells and transplanted them into conditioned wild-type recipients, the donor source is displayed at the bottom. So from the actin line where all the progenitors are present, you can reconstitute the thymus or muscle or other tissues in a dose-dependent fashion. If you take endothelial progenitors, you can reconstitute large vessels. These are the cerebral vessels, or the intersegmental small vessels in the trunk. If you take lymphoid-committed progenitors, they only go to the thymus. So they are definite for their lymphoid commitment. Now, if you do the same study, but instead of taking the GFP-positive cells to adult fish, you actually take them to zebrafish embryos at the blastula stage. The concept here is that if you label an adult progenitor with GFP and you transplant them into the embryo, would they act like an embryonic stem cell? Would they acquire the embryonic stem cell potential from the microenvironment in the embryo? And if this concept proves true, then you can actually do the same thing, but transplanting these cells in mutant fish like P53 or RB, and directly test the effect of these cells in vivo in an animal. So we injected those GFP-positive progenitors at the blastula stage. If you use PBS only, you don't see any GFP expression. If you use cells from the SCL GFP, the hematopoietic stem cells, 2 hours post-embryonic transplant, these cells will start mobilizing. By 6 hours, they are at the hematopoietic sites, and by 22 hours, they recapitulate the normal hematopoietic sites seen in the transgenic fish that expresses SCL GFP. So this is the SCL, the normal SCL GFP transgenic fish expression at 24 hours. This is a transplantation of adult marrow-derived cells into blastula stage embryo. You can go further and recapitulate a whole embryo. So those actin GFP chimera cells, 200 cells, injected at the blastula stage, were able to contribute to all tissues in this embryo, and this was a dose-dependent effect. So if you use only 30 cells, you only small GFP expression here. And you can do it only when you use a ubiquitous promoter that potentially expressing in those pluripotent cells. A lymphoid-restricted expression only goes to the thymus, and an endothelial-specific expression only goes to the vessels. Interestingly, when you use the RAG2 GFP chimera, you can actually detect GFP expression at 3 days post-embryonic transplant, and this is about 3 days earlier than what you would see from a normal RAG2 GFP expression. So these cells are behaving a bit different than the committed RAG2 GFP in the transgenic line. I would conclude that we developed transgenic zebrafish with genetically labeled cells, and these are valuable tools to study the clonogenic potential of adult and stem cell progenitors. We're using them to study the clonogenic potential of the hemangioblast at the single cell level. We describe evidence for adult marrow-derived progenitors reconstituting the endothelium, muscle, and thymus of an adult transplanted vertebrate. We also demonstrate an in vivo direct evidence for adult cell reprogramming in the embryonic microenvironment. Finally, the leukemia work started during my fellowship at NIH. I want to thank Dennis Heckstein at NIH and all those involved in the work, also all the members of CINJ who are making my transition quite easy. Joe Kramer, a recent member who just joined my lab, and all the collaborators, especially Lin Zahn for his significant contribution to the hematopoiesis field in zebrafish, and for sharing the Casper mutants for the transplant studies. Thank you.